When studying the previous session, we had understood the backbone of the DNA is made up of phosphate. A number of histone proteins, they get arranged in a bead-like manner, and around those histone proteins, the DNA gets coiled tightly. If the DNA is tightly wound around the histone, or if the DNA is tightly packed, transcription will be hindered. Now remember, the R strain bacteria is avirulent, but was it heat killed? It was not heat killed. Hello everyone, a warm welcome to another session on chapter 6 of second PUC, that is molecular basis of inheritance. I am Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session of this chapter, we had discussed about the structure of the DNA, right? Wherein we had studied that the DNA is made up of the nucleic acids. And nucleic acids is nothing but made up of three components, that is the nitrogenous bases, a pentose sugar and a phosphate group. So since we know the structure of the DNA and all very well, today let us study about how exactly is this DNA packed within the cell, especially within the nucleus, and we shall look into what are the different experiments that were conducted in order to prove that DNA is a genetic material. So moving on to today's session, let us talk about the packaging of DNA helix. So we all know that the DNA is made up of thousands of base pairs in length, right? And it varies in different organisms. But some of the organisms are so tiny, but the length of the DNA is 10 times more than that of their body size. Can you imagine if the DNA was in a linear form like this, at a stretch if it was there, how would it fit into the body of that particular organism? The body size would have not been sufficient for the DNA to sit inside, right? So that is where the packaging of the DNA comes into help. So the DNA actually packs itself inside the nucleus, and however big the length of the DNA might be, it can easily accommodate inside the nucleus. So how does this packaging occur? Let us look into. So taking the distance between two consecutive base pairs as 0.34 nanometer, that is, it is about 0.34 into 10 to the power 9 meter. If the length of the DNA double helix in a typical mammalian cell is calculated simply by multiplying the total number of base pair with distance between two consecutive base pair. That is, say for example, if it is 6.6 into 10 to the power 9 base pair into one more, that is 0.34 into 10 to the power minus 9 meter per base pair. That is, what is base pair? The number of nitrogenous bases that are present in the DNA, if we count, we'll get the base pair of the DNA. So now, therefore, after this calculation is done, we can find out that the length of the DNA is about 2.2 meters. It's not just the length of the DNA, but also the distance between two consecutive base pairs, right? So together, if we take overall length, after calculating, we can find out it is 2.2 meters. But the cell is so tiny. 2.2 meters, if you see in a scale, you can see 2.2 meters, right? But the cell is so tiny, which is not visible to our eye. To look at the cell and the nucleus, we need to look it under the microscope, right? But imagine how can a 2.2 meter length of DNA, that is a mammalian DNA, sit inside the cell of an organism. So a length that is far greater than the dimension of a typical nucleus, that is approximately 10 to the power minus 6 meter. So we cannot even imagine that this length of 2.2 meters of DNA can sit inside a nucleus, which is far bigger or lengthier than that of that very small nucleus. So how does the DNA pack itself? So in prokaryotes such as E. coli, so E. coli is a prokaryote, it is a bacteria which lacks a definite nucleus. Though they do not have a defined or a definite nucleus, the DNA is not scattered throughout the cell. So how are the DNA packed in the case of prokaryotes, that is bacteria? Prokaryotes, as we know, if I draw the structure of a bacteria, that is a rod-shaped bacteria, the bacteria doesn't have a proper nucleus. Instead, the genetic material will be just like this. It will be scattered at the center of the bacterial cell. It's not that even though the nucleus is not present or a definite nucleus is not present in the case of a bacteria, the DNA doesn't appear in a linear manner. It is always arranged and packed in a coil manner at a particular position here. So, DNA that is being negatively charged is held with some proteins. Why is DNA negatively charged? Because when studying the previous session, we had understood the backbone of the DNA is made up of phosphate, and phosphates are negatively charged. So that is why DNA is negatively charged. Now this negatively charged DNA is held by certain proteins. So these proteins are positively charged, and that particular region where it is held is called as the nucleoid. So the DNA in a nucleoid is organized in large loops held by proteins. How are they held by the proteins? So the best example for the proteins are the histones. They are called as the histones. So these histones, which are the proteins, are positively charged. The DNA is negatively charged because the backbone of the DNA is made up of phosphate molecules which is carrying a negative charge. Therefore, the negative, positive, they attract, and therefore the coiling of the DNA around the protein takes place. So that is how coiling occurs. So studying further, so I gave the example of histones here. So histones are usually the proteins found in eukaryotes, but bacteria or prokaryotes, they'll have a different kind of a protein there. So in prokaryotes, we learned that though the prokaryotes, for example, the bacteria, do not, such as the E. coli, do not have a definite nucleus, but in spite of that, the coiling of the DNA around the proteins take place. Therefore, the DNA doesn't appear in a linear fashion, whereas it accommodates itself inside the cell of the organism at a particular position properly. Now talking about eukaryotes, how is the DNA packed or organized inside a eukaryote? So in eukaryotes, this organization is much more complex when compared to the prokaryotes. So in eukaryotes also, there is a positively charged basic protein which is called as the histones. So here, what are the proteins involved in eukaryotes? Here it is histones. Histone proteins are positively charged, and the protein acquires a charge depending upon the amount of amino acid residues with the charged side chain. So how much amount of positive charge this particular histone protein acquires, it always depends on the amount of amino acids it is made up of. So therefore, histones are rich in basic amino acid, and because it is rich in basic amino acid, it is highly positively charged. So it is rich in basic amino acid residues such as lysine and arginine, and both the amino acid residue, they carry positive charges in the side chain. So histone, if you can see here, this is the histone molecule that they have shown. So this histone molecule, it is a octamer. So this histone molecule is octamer. Octamer means it is made up of 4 different components, and these 4 components are arranged in 2-2 pairs. So therefore 4 2s are, it will become 8, right? So that is why it is called as a histone octamer. So this histone octamer is a protein. Histone is a protein. What are the charges that are present on the protein? It is positively charged, positive charge, right? Why is the histone positive charge, or how much amount of positive charge the histone carries depends on the amino acid residues it is made up of. What type of amino acid residues? It is made up of the residues such as lysine and arginine. So lysine and arginine both carry positive charges. So therefore, this particular protein is positively charged. And we all know that proteins are nothing but they are made up of chains of amino acids, right? So that is why. So therefore, the histone is positively charged, and these histones are organized to form a unit of 8 molecules called as the histone octamer. I told you there are 4 components present in the histone, and these 4 components are each in 2 in number. Therefore, 4 2s it will become 8. It will become an octamer. Octa means 8, right? So therefore, it forms a histone octamer, and the negatively charged DNA. Why is the DNA negatively charged? Again here, because the backbone of the DNA is made up of phosphate. The phosphate is negatively charged. Therefore, the negatively charged DNA gets wrapped around the positively charged histone octamer to form a structure called as the nucleosome. So that is how the nucleosome. What is a nucleosome? Nucleosome is nothing but the content that is present inside the nucleus. What is it made up of? It is made up of DNA, chromosomes. In the chromosomes, what are there? The DNA is there. The DNA is wound around the histone protein. So all these together, they form the nucleosome. So this was about DNA packaging. So these nucleosomes, they constitute the repeating unit structure. So now, is one histone octamer or histone protein enough for coiling a very big 2.2 meter length of DNA? Not possible, right? So therefore, a number of histone molecules will occur. So if you can see here, you can see this DNA. This is the DNA strands, and these are the histone proteins. So a number of histone proteins, they get arranged in a bead-like manner, and around those histone proteins, the DNA get coiled tightly. Because of that coiling, the lengthy DNA accommodates itself very neatly inside the nucleus. So here nucleosome constitutes the repeating unit of a structure called in the nucleus, which is called as the chromatin. So what is this chromatin? So together, all these together, we call it as the chromatin. So again, what is there inside the nucleus? Inside the nucleus, it is made up of nucleosome. What is there in the nucleosome? The nucleosome is nothing but chromatin. When you clearly observe the chromatin, it's nothing but chromosomes. Those chromosomes are nothing but the DNA which is wound around these histones. So therefore, these chromatin, which are thread-like stained bodies seen in the nucleus. So chroma means color. So therefore, they are stained bodies that are present, or colored bodies that are present inside the nucleus. And a typical nucleosome can accommodate about 200 base pair of DNA helix. So, and the nucleosome and chromatin are seen as beads on a string structure when viewed under an electron microscope. So, if you see them, they almost look like a bead. So, how you have a chain, right? When you wear a chain, how it looks, exactly like that, they will look like a bead, as if it is a string of beads. So, that is how the DNA gets packed, and the beads on a string structure in the chromatin is packaged to form the chromatin fibers, and they are further coiled and condensed at the metaphase stage. That is why it is in the metaphase stage that we can clearly see the chromosomes, because these histones furthermore coil the DNA properly, and therefore the chromosome gets reduced in size and it becomes more thicker. Therefore, it is easily visible in the metaphase stage of cell division, wherein the actual structure of the chromosome can be visualized. And the packaging of the chromatin at higher level, it requires additional set of proteins, and these additional set of proteins are referred to as non-histone chromosomal protein. So apart from this, as the hierarchy of the organism increases, or as the complexity of the organism increases, more coiling of the DNA needs to occur, wherein extra proteins has to take part, and such proteins are called as the non-histone chromosomal protein. So this is how the DNA is packed. So how does the DNA packaging occurs? Here the DNA, no matter what the length of the DNA is, the DNA can accommodate itself in the nucleus, because the backbone of DNA is made up of phosphates, which is negatively charged, and these negatively charged phosphates, they bind themselves around the positively charged histones, and therefore they form a string-like structure. And it is because of this coiling around the histones, any length of DNA can accommodate itself inside the nucleus properly. And apart from that, there are also certain, in higher organisms, as the complexity increases, there are other proteins which are called as the non-histone chromosomal proteins that also participate in the packaging of the DNA. So in a typical nucleus, some region of the DNA are chromatin are loosely packed. So here the packing of the DNA around the helix or around the histone, or the coiling of the DNA around the histone can be very tight or it can be a bit free. So here in some of the that is in typical nucleus, some region of the chromatin are loosely packed and are referred to as euchromatin. Because they are loosely packed, they are referred to as euchromatin. And the chromatin that is more densely packed or tightly packed, they stain darker and they are called as heterochromatin. So therefore, it is the heterochromatin that we can observe during the metaphase stage. And euchromatin is said to be transcriptionally active. So what it means that if the DNA is tightly wound around the histone, or if the DNA is tightly packed, transcription will be hindered or transcription might not occur. So what is transcription and all that, you'll study in the coming sessions, wherein you have topics on that. So here, and on the other hand, because heterochromatins, the DNA is tightly wound around the histones, or the DNA is tightly packed, the transcription process will not occur or it will get hindered. But in the case of euchromatin, the coiling of the DNA around the histone is not that tight. It is loosely coiled, and loosely coiled DNAs actually facilitate the transcription process. So therefore, euchromatin is said to be transcriptionally active chromatin, whereas heterochromatin is transcriptionally inactive. So this is also very, very important. So therefore, wherever in the chromatin the DNA is packed loosely, their transcription will occur. Wherever in the chromatin the DNA is packed very tightly, the transcription will not occur there. So this is about the packaging of the DNA. So therefore, we'll study, now we know about the packaging of the DNA. In the previous session, we understood the structure of the DNA, that is, it is a double helix model and all that we had studied. So now we'll study how exactly the search for genetic material came into existence. Because Mendel, during his experiments on the garden pea plant, he had proved that there are some factors which carry the information or carry the messages or carry the characters from the parents to the progeny. That is why the progeny has some of the similar characters to that of the parents. But he didn't know them that it was genes and it was DNA that was responsible for that. But how did other scientists find out that actually it is DNA that is the genetic material that, so what Mendel called as factors is nothing but genes. Those genes are present in the DNA, and therefore DNA is the genetic material that carries the information from the parent to the offspring. So therefore, how did exactly scientists come to know about it? That DNA is a genetic material we shall look into. We shall study about a principle called as transforming principle. So in 1928, a scientist, that is Frederick Griffith, he conducted a series of experiments using Streptococcus pneumoniae. So this is a bacteria that actually causes the pneumonia disease. So he was conducting experiments on that, studying the growth of that bacteria and all that. And suddenly one day he witnessed that a miraculous transformation in the bacteria was found. That is, in the texture of the bacteria. Some of the bacteria were rough in texture, some of the bacteria were smooth in texture. So keeping that in mind, he conducted few experiments. What was that? We shall study. So during the course of his experiment, he used a living organism, that is bacteria, which had changed in physical form. So this discovery was actually accidental because he had left some plates just to wash it as such. He had left some plates to discard or something, and one day when he went near the culture plates and he saw that those bacteria, that is Streptococcus pneumonia, they showed some variations in their outer texture. As I told you, some were very shiny and smooth, some were very dull and rough. So he wanted to understand why is that variation, why did that sudden variation occur. So that is when he used that Streptococcus pneumonia to study to conduct various experiments. So what he did was, when he grew Streptococcus pneumonia on the culture plate, that is, when Streptococcus pneumonia bacteria was grown in a culture plate, he found out that again, whatever culture plate he had left, no, to throw it away, to wash it off, again from there only he took the bacteria and he grew it again in a fresh culture plate. And again he observed that some produced smooth shiny colonies while other bacteria, again other bacteria means in that particular culture plate only, some of the Streptococcus bacteria, they produced rough colonies. He wanted to understand why was that. So that is when he started doing the experiments, and he found out that this was because the S strain bacteria, they had a mucus around it. So wherever there is mucus, we know that it appears shiny, right? Because mucus is nothing but a slimy gelatinous substance. Therefore, that bacteria, that is S strain bacteria. So why did he give the name S strain? Is because it had smooth shiny surface. For rough surface, he gave the letter R. He designated the bacteria as letter R. So here this is because the S strain bacteria, it produced a mucus around itself. So the mucus was nothing but it was made up of a polysaccharide. So because of that particular mucus coat, the bacteria appeared to shine. Whereas the R strain bacteria, that is the rough strain bacteria, did not produce any polysaccharide mucus coat around it. Therefore, they appeared rough. They did not appear shiny. So why was this? What exactly is the reason behind that? So what he did was, he took those two strains to find out, will those two strains show some similar characteristics or not? Whether, because whenever we talk of a disease, we talk of its symptoms, right? So he wanted to understand whether this smooth stained Streptococcus pneumonia bacteria causes the same symptom and the rough Streptococcus pneumonia bacteria causes the same symptom. So to find out the symptoms, what usually scientists do, they injected it to a mice, right? So that is what he also did. So what he did was, he injected the mice with S strain. So S strain, he considered it as virulent strain. So what is S strain? It is having a mucous membrane around it, which is made up of a polysaccharide, right? So that S strain he injected into the mice. So when he injected, after a few days, the mice died from pneumonia infection. And so he took two sets of mice, okay? So one set of mice, he injected it with those Streptococcus pneumonia bacteria which were shiny. And another set of mice, he injected with those Streptococcus bacteria which were rough, which was not shiny. So those mice which were injected with Streptococcus pneumonia having shiny smooth surface, those mice died because they got infected by the pneumonia. But on the other hand, those mice which he had injected with the rough strain Streptococcus pneumonia bacteria, they didn't die, they lived. That was the findings he actually got. So therefore, those infected with R strain did not develop pneumonia and they tend to live. So that is what he did. What he did, he took the S strain Streptococcus pneumonia, he injected into a group of mice, those mice died. And on the other hand, he took a rough strain Streptococcus pneumonia bacteria, he injected into the mice, but the mice lived. Why? Because these were infected with pneumonia, but these did not get any infection with the pneumonia. So that was the reason. So next what he did was, again he conducted an experiment. So S strain is virulent. So in biology you need to know about two important terms. One is virulent and the other one is avirulent. And I told you, whenever the word A comes in front of some other word in biology, it means it is not exhibiting any property. It's like that, or it is absent. So virulent, which was the virulent bacteria? Virulent bacteria are the ones which are highly active and are capable of causing a particular infection. So here the S strain Streptococcus bacteria is virulent, whereas the R strain bacteria is avirulent. It will not cause any infection. Now what he did was, this S strain, How can we kill the bacteria and make it avirulent, make it inactive? Virulent means active. Again, here I'll write this for you to easily understand. Avirulent means consider this to be inactive. Now, he took the S strain and he wanted to make it inactive so that it will not cause a disease. So, how did he make it inactive? He killed the S strain bacteria by heating them. So, what did he do? He killed the S strain bacteria by heating them, and he found out that the heat killed S strain bacteria. Now, in his mind, he had that, okay, I've killed that S strain bacteria. Now, that S strain bacteria is inactive, means it is avirulent. He injected that avirulent S strain bacteria or the heat killed S strain bacteria, he injected it into the mice, and the mice did not die. Why? Because the S strain bacteria was heat killed. When it was heat killed, what happened? It became inactive. Therefore, because it became inactive, when it was injected into the mice could not develop pneumonia and the mice lived. So, that is what he observed. And next what he did was, again he was not satisfied. What he did was, he injected a mixture of heat killed. Now, he took a small beaker, in that he poured some solution. In that solution, he added the heat killed S strain bacteria, and he also added the R strain bacteria. Now remember, the R strain bacteria is avirulent, but was it heat killed? It was not heat killed. S strain bacteria is also avirulent. Why it became avirulent? Because it was heat killed. Now he mixed the heat killed S strain bacteria with the avirulent R strain bacteria, and those two together he injected into the mice. So, when he injected a mixture of heat killed S strain bacteria and the live R bacteria, the mice died. Now, he was in a shock. How did the mice die? The mice were supposed to live, right? Why? Because the R strain bacteria is avirulent. It was not supposed to cause infection. That was in his first set of experiment. He found out that, right? R strain mice lived, but in this particular case, they died. So, he got more enthusiastic, curious. He wanted to know why did this happen. So, what he did was, he again recovered the S bacteria from the dead mice, and then he conducted studies on that. So, what was his experiment? First, he took the S strain, he heat killed the S strain, then he injected the S strain into the mice, and he noticed that the mice lived. Why? Because this bacteria became avirulent. It became inactive. It lost its capability to cause the pneumonia infection. So, therefore, next what he did was, he again took the S strain bacteria. This is also avirulent. And he took the live R strain bacteria, because R strain bacteria, there was no need to kill the heat kill the R strain bacteria, because first of all it did not cause infection in his early experiments, right? So that is why. So this was also avirulent. What he did? He injected both of those into the mice, but the mice which was supposed to live, it died. So he got curious. So therefore, what he did? He again cultured this S strain bacteria, not cultured, he again recovered the S strain bacteria from the mice, and he concluded that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. If you remember, I had told you the S strain bacteria is shiny, smooth, right? Why is it shiny, smooth? It's because its external surface was covered by a mucous membrane that was made up of polysaccharide. So, something from that had transformed into the R strain, which was living. Though it was avirulent, it was living. So, that is why, so this is nothing but the transformation principle. So, transforming principle wherein the something from the S strain, that is from the heat killed S strain, transferred to the R strain, and therefore the R strain started to synthesize a smooth, shiny polysaccharide coat. So now what happened here? Because the S strain was heat killed, It was inactive. But was the R strain inactive? R strain was live, it was living because it was not heat killed. But R strain is avirulent, not capable of causing the disease. But when he mixed both and injected it to the mice, died. Why was it? Because the polysaccharide that was present on the mucous membrane of the estrain bacteria, that did not get altered even though the bacteria was heat killed. So from that gene that is responsible for the for making this particular polysaccharide got transferred into the living R strain bacteria, and now the R strain bacteria became virulent, that is gained its capability to cause pneumonia disease. So that is why when he injected it into the mice died at the end. So this must be due to the transfer of the genetic material. What genetic material? That particular gene that produced that particular polysaccharide membrane got transferred from the S strain to the R strain. Because the S strain was not living, it could not express. R strain was living, therefore, though it was virulent, it got converted into virulent and it started to cause the pneumonia disease. So, however, this biochemical nature of genetic material was not defined from his experiments, but still people were not satisfied with his experiments. But he was the first person, Frederick Griffith was the first person to say the DNA is the one. So DNA is he did not know it is DNA and all that, but he knew that the genetic material is there. It gets transferred from one organism to the other of the same species, therefore causing a lot of changes in that particular organism. So this was his particular experiment. So this is one small experiment that led to the discovery of the genetic material, or for the search for the genetic material. Other than this, there are other experiments as well which we will study in the coming session. So in the coming session, we shall again continue with studying the search for genetic material and how eventually scientists found out that the genetic material is nothing but DNA. So the first step for the search of genetic material, the credit goes to Frederick Griffith because of his experiments conducted on Streptococcus pneumonia bacteria. So this was about this session. So I hope you understood this session. We shall meet again in the coming session. Thank you.